thanks very much. I am Justin Bohn from Johnson & Johnson's epidemiology group, and I'm going to talk to you today about some work we published in Drug Safety recently called Large-Scale Empirical Identification of Candidate Comparators for Pharmacoepidemiological Studies. So first, I'll say a bit about the traditional approach to comparator selection. So the new user cohort design, as most of you know, has emerged as a best practice in pharmacoepidemiology, but it requires the selection of an appropriate comparator for whatever target outcome relationship you're trying to study. This can be partially subjective and often is done on the basis of what other people have done or published on, what alternative comparators are available in your preferred data source, what gives you the biggest sample, what you think might happen with confounding, and then if you're a bad epidemiologist, you might choose the one that you think will give you the effect that you want. The actual comparison of interest could also be to a non-user group, and this is a challenging scenario, but you can emulate it in the new user cohort design using what are variably called negative control exposures or null comparators. And I would note that this situation is often implicit in safety evaluations, even when we know better than to say so as investigators. So the motivation for this work is basically, can we provide the investigator with some empirical information to help them make their comparator choice? And our proposed solution is to build a tool that identifies new user cohorts that have similar covariate profiles to each other to enable comparator selection and identification. This involves defining new user cohorts in real-world data, extracting covariate information on those cohorts, computing similarity metrics between pairs of cohorts, ranking potential comparators, and then the ability to actually explore that comparison. We evaluated the performance of the tool that we created to do this based on whether highly ranked comparators that we generate from it appear clinically relevant. Also, whether the computed similarity aligns well with known relationships like those in the ATC hierarchy, whether the computed similarity corresponds to more familiar measures in the pharmacoepidemiological context, like the standardized mean difference, and whether the tool tends to rank highly those comparators chosen by experts in high-quality publications. So in terms of methodology, to build our pipeline, we start with the generation of new user cohorts for thousands of drugs and classes, leveraging the pre-computed drug eras from the OMOP CDM and imposing a 365-day prioritization requirement and washout, and then repeating this process for each of the Rx norm ingredients and ATC level 4 classes that are in routine use in our data sources. We used 5 data sources that are at the bottom right here. Once we have those cohorts generated, we have a data adaptive covariate data extraction process. Custom SQL aggregates or extracts aggregated pre-treatment covariate data in these 5 domains here. So we have demographic data and then data adaptive domains related to recently diagnosed conditions, recent visits, more distal conditions, and more distal medications recorded. These are designed to emulate clinical decision making, and I would note that we remove rare covariates with a prevalence less than 1% in order to avoid skewing the similarity calculation later on. This is pretty similar to what's done in the feature extraction package, but it's a little bit faster and lighter weight. The next step is actually the similarity calculation, which starts by forming all pairs of new user cohorts with at least 1,000 patients. We compute a cosine similarity score for each pair separately in each of those 5 domains that I just showed you. You can see the formula right. And then we take the average of the cosine similarity scores across those domains to get what we're calling the cohort similarity score, the final similarity score. And we can use that to rank comparators for any given target. So I'll get into the results a bit now. First of all, I'm just going to show you some top-ranked comparators for 6 different drugs from 3 different therapeutic areas, and I'll focus on the right 2 columns that are drugs for multiple sclerosis. So these are the 5 most similar scores, 5 most similar comparators by average cohort similarity score across databases. And for these 2 multiple sclerosis drugs, glatiramer and ocrelizumab, you can see, if you're familiar with the space, that these are all either disease-modifying therapies for multiple sclerosis or symptomatic therapies for multiple sclerosis. So this method identified these drugs with no embedded knowledge of what the indications for the drugs are, or any use of any drug hierarchy. It's purely based on similarity in the high dimensional covariate space. And you can see this is the case for the other examples as well, that this is consistently producing clinical alternatives and related treatments for each target. We also benchmarked this method against a more familiar pharmacoepidemiological metric of covariate similarity, the standardized mean difference. And the way we did that was to take 100 randomly selected target cohorts in each database, identify the top ranked comparator, number 1, and a bad comparator, ranked number 500, compute the standardized mean differences for all the covariates in the comparison within each of those cohorts, and graphically depict the distribution of absolute standardized mean differences to you here using standard cutoffs for degrees of imbalance. And you can see pretty clearly that if you choose a top comparator, you're getting a majority of covariates that are already balanced according to the standardized mean difference being less than 0.1. Whereas if you choose a lower ranked comparator, you're going to get quite a bit of covariate imbalance. So this is reassuring based on some more familiar metrics of covariate similarity. Okay, and we also performed a literature review to identify how well our method ranks comparators chosen by expert opinion. And we did this by performing a search for real-world data analyses that use some keywords to identify new user cohort designs in recently published papers in high-impact clinical and pharmacoepidemiological journals. We identified 60 such papers, of which 24 were actually employing a new user design, and it represented 56 comparisons, of which 23 could be mapped completely or indirectly to either ATC or RX norm. And among those pairs, in at least one data source, the chosen comparator was in the top 5% of similarity for the target in 96% of cases, was ranked in the top 10 in 91% of cases, and was ranked in the top 5 in 78% of cases. So in general, the method tends to rank highly those comparators chosen by experts using their subject matter knowledge, presumably. This is, again, reassuring. I will also note that along with this evaluation, we have an interactive web application where you can select a target exposure, you can select some setting values for the comparators you want to see, and receive a ranked list of candidate comparators, which you can explore interactively. You can obtain plots of covariate prevalence, standardized mean differences, table 1 type tables of covariate distributions that are searchable. And so you can find out some information that's helpful to you, including, not shown here, but there's also functionality to identify covariates you might wish to exclude because they're very different between the target and comparator cohorts, which can help a lot with your propensity score analyses. This is available at data.odyssey.org. You can use the QR code, or if you can read that really small text for the URL, you can type that in. It's also linked in the paper. So there are, of course, some limitations to this approach. This methodology doesn't account for the covariate importance, so it looks at basically balance between target and comparator cohorts, but not their importance in terms of outcome relationships. And it doesn't say anything about the joint distribution of covariates, which something like a propensity score might do. Also, it should be noted that the similarity in high dimensional covariate space between a target and a comparator doesn't necessarily imply the adjustability of those that comparison in a final analysis. But there are plenty of potential mitigations here. This is probably most useful as a screening tool before which you apply something like a large-scale propensity score analysis. The recommendations themselves are also sometimes problematic for drugs that are combination products or have multiple potentially disparate indications. If anybody wants to know more about that, I have some backup information about that that you could ask about later if you wanted. And drugs that are used differently depending on dose, form, or route of administration. This can be mitigated by the addition of custom cohort definitions, running the pipeline in your own data, or running the pipeline nested within an indication fully. So in conclusion, we developed a novel tool for assessing the similarity of new user cohorts at scale and in high dimensional covariate space. the comparators coming out of this tool appear clinically relevant, align well with choices made by experts, and already produce balance on the majority of covariates with respect to the target in many cases. This is a continuing process. We're continuing to improve this tool and welcome any questions you might have now. Please read the paper as well, and we'd love to hear from you. Thanks very much.
Great, thank you, Justin. We have about 3 minutes for Q&A. We're fast here. Patrick, go ahead.
Yeah, I'm just gonna thank you, Justin, for presenting this, and I appreciate that you've been bringing this to the community. I just wanted to share with you and the group. We actually ended up teaching the comparator selector tool at our Odyssey summer school last week, and there was a bunch of excitement from the students who used it, and there was actually 2 use cases that I thought were interesting to highlight that I think you touched on briefly, but one was not one I expected. So there was an interest in a particular drug, and the student didn't know what the right comparator to choose was. So that was the use case that you kind of highlighted. They typed in the drug, they saw the candidates at the top of the list, and they were able to basically like sort through the rank ordered list and find one that was good. But then by reading papers, they saw a paper that took that same drug and made a comparison to another drug. And so they used the comparator selector to basically ask the question, was that a good comparator? And they saw that that comparator drug was actually like 207th in the list. And it did 2 things. One is it said, well, if there's 206 drugs that are probably better comparators, maybe we should think about that. But also because of that nice drill down feature that you showed us, we were able to basically say, like, why is this drug that somebody else had published that they picked this comparator maybe not such a great comparator, and was able to immediately expose the difference that was not immediately obvious to us as readers. So I just want to, one, commend you for the tool, know that we're using it in our education and encourage others to use it. But also I think that there's multiple different angles we can go at to use this resource, and including ones that we as co-authors of the paper didn't necessarily think of immediately, but turned out to be very useful.
That's great. Great to hear.
Any other questions?
I agree with Patrick. The tool is very neat, and you could go beyond research. I mean, there are a lot of interesting things about 2 drugs being used in similar situations. If you're stumped about what drug to give next, I mean, in theory, you could come on here just out of curiosity and just see what other people are giving. You would never just give a drug just based on it being high on this list, but it's information that could be useful.
Yeah, for sure. Thank you very much, George.